Okay, tonight's video is going to be on DNA replication and answering the question, how does DNA copy itself? So if you're going to make new cells or repair cells, how are you going to make copies of DNA? So just as a review from the last section that we went through, some of the base pairing, um, A and T pair up together and G and C pair up together. They make up the genetic code, with A, T, C, and G being the alphabet. These base pairs in the code can be translated into instructions to make a protein. They can make enzymes to speed up or to help out in a reaction. And the overview of what DNA is, it's the instruction manual to make proteins. So here's an overview video that you can watch on replication process. It's going to take a second to click on that link and go over and watch that video. And just as an overview of replication, you're going to start with one DNA molecule and end with two. So that's your starting products and ending products. The why of doing it, it's important because each cell at the end of mitosis is going to need an exact copy of the instructions it needs to carry on the processes for life. So you can't create a new cell without it having the instructions and be able to do all the process it needs to. So you're going to need to copy DNA to send over to that new cell. There are 4 enzymes that you're going to need for replication: helicase, DNA polymerase, ligase, and primase. We're going to go over those 4 in more detail, but I've got a link for a Quizlet page that's got the enzyme and the definition. So if you want to practice those, you can click back over and practice those on Quizlet. Okay, so the first step, you got to think your DNA strands this wound up ladder. First thing you need to do is unwind it. So replication is going to begin at a specific site in the DNA called the origin of replication. So that's like the marker spot for where replication is going to begin. And then replication is going to proceed in 2 directions at the same time. So once we get that DNA unwound, DNA is going to be replicating on this side. So one newly forming strand will be here, and one newly forming strand will be on this side. DNA helicase causes the 2 parent DNA strands to unwind and separate. So we've got a replication origin. DNA helicase attaches on. It starts to spin and unwind the DNA, causing this split here. And that area where replication is happening is called the replication fork. So second step is we need to start building our new DNA strands. New nucleotides are going to line up opposite the parent strand. So we've got the original strand. If you look over at the picture here, you've got the original strand, and then you've got the one that's newly being built. DNA polymerase joins the new nucleotides by their phosphate groups. So if you remember, this outside part here is sugar phosphate backbone. Polymerase joins the new nucleotides by the phosphate groups, and then the phosphates that are removed provide energy to drive more binding of the nucleotides. And the nucleotide is the 3 parts: the phosphate, sugar, and the base pair. Step 2 continued. If you looked back on this slide here, DNA polymerase is going to move in one direction, which is fine for the one strand. It's just going to, as helicase unwinds going this way, polymerase is going to keep building in this direction. But for this top part, it creates a little bit of a problem. Polymerase is only moving from left to right here in this lagging strand, which means that it's going to have to create, on the lagging strand, DNA is going to be made in installments. So it's going to take 1,000 to 1,000 nucleotides at a time and build nucleotides in that direction. These newly formed fragments are called Okazaki fragments for the scientists who discovered them. And another overview, so after you've reviewed the vocabulary for the different enzymes, and you look back and you understand each one of the slides, you can go to this link. It's got a bunch of good information on DNA replication.